Hi everyone, my name is Lindsay Rathbun. I'm an MCDB major and I'm graduating in the spring of 21. I was in the Friday lab, section 16, and today I'll be presenting my poster, Tetrahymena thermophilia HMG box protein inactive in DNA damage response pathway. The DNA damage response by cells is utilized so many times throughout the day, thousands of times, but it's mostly unknown the mechanisms behind it. When this pathway is damaged or inactivated, it can cause issues such as mutagenesis or even cancer. If those damaged and mutated cells are allowed to persist, it can pass the mutated traits down to the next organism's generation. By learning more about this response pathway and its processes, it can allow us to eventually develop treatments. In this experiment, we use Tetrahymena thermophilia, a unicellular eukaryote, as seen here in the introduction, that repairs its own DNA during mating. By inducing DNA damage with hydroxyurea, which causes double-stranded breaks in DNA, we can look at the cellular response to the DNA damage by looking at the resulting gene expression. If the gene expression increases, then we can likely suspect that the gene was recruited and is involved in the DNA damage response pathway. My gene, T-therm005680, is a high mobility box protein that is likely involved in DNA binding. For our first PCR, we used RPPO primers, which is a known gene present in Tetrahymena, and we were able to see that it was successfully amplified with the expected gene fragments, and that validated our ability to first run a PCR. And then we used gel electrophoresis to visualize the resulting DNA fragments. After confirming that PCR, we then designed gene-specific primers to amplify our gene of interest. And we identified any homologs and other model organisms. In our second PCR, we confirmed that the designed gene-specific primers were able to anneal properly to our gDNA and cDNA, as seen in the first figure below. And after confirming that they were able to bind, as seen by the bands present at the 0.5 kilobases in the figure, we were able to receive log phase hydroxyurea treated cDNA to run our experimental PCR. We used RAD51, which is a known gene that is involved in the DNA damage response system. So we expected that the gene expression would increase, which it did, as seen in lane 2. In fact, the cDNA band in lane 2 was about 4,000 times brighter than the band in lane 1, indicating that it was recruited in the pathway to repair the damaged DNA. The RPPO bands indicated that the DNA was damaged because in lane 4, the hydroxyurea treated band was dimmer, which indicates that the DNA was damaged because it's a known gene in Tetrahymena, but it's not known to be part of the DNA damage response system, so it was only damaged by the hydroxyurea and not able to repair itself. And finally, in lanes 5 and 6, you can see that there are no noticeable bands visible to the naked eye. And after quantitative analysis, it was determined that there was actually a 0.5% decrease in gene expression. This indicates that my gene is not involved or inactive during the DNA damage response pathway utilized by the cell. While it can be concluded that my gene of interest is not involved in this vital pathway, that does not mean that the research should stop here. There is still much information to gain from studying this topic by using other candidate genes. And it can help us develop treatments in order to intervene and either stop the damaged cells from persisting or repair the pathway altogether. Thank you so much.